Good morning, everybody. We are going to wait just a minute or two and let a few more people filter in. So thank you for joining us this morning. We are really excited to share about one of our most important programs with you. We will probably get going in about 2 minutes, so maybe one. So finish answering some emails and pour yourself another cup of coffee, and we'll be right with you. It's great to see a lot of familiar names as you all are joining. For those who are just logging in, we're going to just give it about 1 or 2 more minutes, let a few more people join, and then we will be on our way. Okay. All right. Well, I will start with some quick brief introduction to our purpose here this morning and the folks that I have with me here in this virtual room. First, I want to say thank you all for finding a little bit of time to spend with us today, whether it's morning or evening. We are really delighted to have you with us to talk and learn a little bit more about our International Multi-Location Variety Trial. This is one of WCR's most important programs. It was really the first program we launched, the big program that we launched after we were formed in 2012. And we're really excited to share with you some of our kind of early global results in this webinar this morning. Before we dive into all of that, I just want to make a couple of introductions. I'll introduce myself first. I'm Hannah Neuschwander. I am WCR's Director of Communications and Strategy. My role is to help explain what we do and why it matters. So hopefully we are on track to do that this morning. I also have with me this morning Dr. Tanya Humphrey, who is our Director of Research and Development. Tanya really oversees all of our scientific programs, all of our trials, our breeding network, and is going to be sharing some of the data and results from our IMLVT trial, our International Multi-Location Variety Trial today. I also want to give a special shout out to Catherine Mena, who is our Global Research Project and Product Manager. Catherine's based in Kenya, and she is really the glue that holds this entire thing together. Catherine oversees all of the operations and logistics behind this trial, which, as you'll learn, involves 28 sites in 19 countries. So it is a very large, very complex program, and Catherine is the person who keeps it all together and makes sure that we're moving forward. So without her, this really wouldn't be possible. I also want to introduce Dr. Jorge Berni, our research scientist for plant breeding and genomics, and all of the data that you see here, all the tables and charts and beautiful graphics that represent the performance of these plants all over the world. All of that comes from Jorge. He does all of the data analysis for this trial and figures out how to take a lot of numbers and raw data and turn them into meaningful information that we can use to make decisions to help drive global production forward. So thank you guys so much for joining me this morning. You can go ahead and turn your screens off while I do a couple of slides, and then we'll bring folks back in for looking at the data and for some Q&A. So I'm going to give a little bit of background about what this whole trial program is, what the International Multi-Location Variety Trial is. I'm going to go ahead and just call it the IMLVT. That's our funny acronym for this. It's a little faster than saying the whole thing. So IMLVT just means International Multi-Location Variety Trial. And then Tanya's going to come in and share some of our global results with us, and then we're going to have some opportunity for questions. Before I dive in, I just want to pause really quick and offer a thanks to our member companies, in particular our global sponsors. This trial, as I mentioned, has been going for since about 2015. It was really the first big project that we got off the ground. And so it is a multi-year project that has required multi-year funding, and there is no way that we could do this, something this ambitious, without the support of our member companies. So we really appreciate that. So as most of you know, I think, one of the primary things that we do here at WCR is we focus on variety innovation. And the reason that we focus on variety innovation is because it has tremendous potential and power to affect multiple sustainability and business challenges at the same time. When you have a new variety that is, for example, tolerant to key diseases and pests that a farmer might face, and is higher yielding than the other ones the farmer might have access to, and is able to, survive through climate variability, hotter temperatures, colder temperatures, drought, frost, etc., and has good crop quality, you have, in just the form of one small little plant, you have contributed substantially to a farmer's ability to remain financially stable and profitable in their farming business, to decrease agrochemical use, decrease pesticide use if it's a pest-resistant plant, potentially to decrease greenhouse gas emissions, and land use conversion and deforestation, because you can produce more coffee on a smaller amount of land. So really, varieties have a huge potential to impact the entire landscape of coffee production around the world. And as a global organization, working on varieties is a very efficient way to invest your dollars. We can really make a lot of change in an efficient way through variety innovation. So it is one of the core things that we focus on, and we focus on it in a few different ways. So we have 4 program areas to ensure that better coffee varieties are making it into farmers' hands. The first is breeding, so actually creating new varieties. Some of you may know that we just launched a large new, it's our newest, biggest program called Innovea, a large global coffee breeding network, which is very ambitious and exciting, but breeding takes a long time. So at the same time, we also work on trials. So looking at existing varieties or new-ish varieties and evaluating and ensuring that they actually do have higher performance for farmers. Because you can create a new variety, it might seem to be good, but you got to get it out into the field and make sure that it really is better than what farmers already have access to. Otherwise, it's not worth their time or money to plant them. And then you need a strong nursery and seed sector in order to facilitate access to varieties for farmers. You can have a fantastic variety, but if a farmer can't get a hold of it, it doesn't matter that you've made it, right? And finally, global advocacy and alignment, because we know that there is a tremendous gap when it comes to coffee agricultural R&D. We, as a global industry, as a global sector, need to be spending somewhere in the area of about $200 million a year on agricultural R&D in order to support this crop. And we're about $100 million short of that, we think. That's our back of a napkin math. So we really need to mobilize support and funding from a variety of sources and have a sort of all hands on deck approach to increasing coffee agricultural research. And so we do that by bringing our member companies together and working with others in the sector to try to drive increased support for coffee R&D on the whole. So I mentioned that, one of the challenges with varieties is that breeding in particular can take a really long time. So because coffee is a tree crop, it can be very slow, the breeding process. So even if you're using the kind of most advanced techniques and technologies, you're looking really at a minimum of 10 years from when you start the process to when you might have something that could go out into pre-commercial trials. It could be as long as 30 years. With most traditional varieties that farmers are growing today had a 30 to even more year breeding process. I was just on a call yesterday where someone was asking me about a Honduran variety called Paranaima, and it was new to this person. His family has a farm, and they were asking about its development. And I said, yeah, that was developed in the 90s and released in the early 2000s. And they were shocked that the variety was that old because it was new to them. And that's very common in coffee agriculture. So breeding takes a long time. And part of what that means, if we want to have an impact today for farmers, is that we also need to do things that have shorter timelines simultaneously with working on breeding. We have to work on breeding because the timelines are long. We have to start now, but we also need to do other things. And so for us in our portfolio of work, that also looks like trials. So that's maybe taking existing varieties and testing them in other conditions or new locations where they haven't been tried before to see if they might have some potential in those locations. And then also nursery work, as we mentioned, just cleaning up the quality of plants that are being reproduced for farmers, and also the genetic purity, making sure that varieties really are the varieties that nurseries think they are, that farmers think that they're getting. It has a much shorter term impact. And then of course the global leadership piece can have a very quick impact if you're able to mobilize increased funding. So in the trials bucket of our work, really the goal here is to fill the shorter term need that we have, given that breeding does take such a long time. And in particular, we have this trial, the International Multi-Location Variety Trial, or IMLVT, which is put in place to help countries, different countries around the world, different coffee producing countries, evaluate and access better current varieties from other locations. So this trial was started, as I said, in 2015. And what we did was we took 31 high performing varieties from all over the world. This is a very small little graphic here, but you can basically see that we have a bunch of countries listed and underneath them a number of varieties. So Pacamara from El Salvador, Geisha from Panama, some F1 hybrids from Central America, Oro Azteca from Mexico, et cetera, et cetera. So we collected all of these varieties and we made a pretty significant effort to go to trusted partners who would have the actual variety, right? Sometimes what farmers have in their fields isn't actually the variety that they think it is. So we went to the breeder or a trusted institution to obtain these varieties, and then we reproduced them. We basically made little clones, little test tube coffee babies at a lab in Florida. And then we sent them back out to 29 sites in 18 countries. Many of the varieties that are in this trial had not been tested in anywhere near this many locations. So a variety like, I'll just give a small example, like Pacamara is very common in El Salvador. You do occasionally find it on farms in other places where people have, enterprising farmers have decided to test it out, but it's not widely available and you generally wouldn't find it in Asia or Africa. And vice versa, there's African varieties that you wouldn't find in Central America. So this was a large, very well organized, systematic trial to take all of these different genetics, these different varieties, and put them in different environments and see how they do. The picture on the left is at the baby stage with our trial in Australia. So you can see sort of how the trial is set up in these very neat rows. Each row is a different variety. They're separated from the other rows so you don't have cross-pollination. And there's a lot of very systematic data collection that happens on each individual plant to see how they're performing. So here's the full list of varieties, 31 varieties, as I said, from around the world. You can see on the picture in the bottom, some little tiny baby plants. So, all these varieties went out as little test tube babies to all the countries. They took them, they had to root them in nurseries and then transplant them to the field. The logistics behind this are really, really tremendous. And of course, not all the plants survive. That's why in the previous picture, you see some empty spots in the row. That's very normal, but it's kind of amazing when you think about the logistics that really we have gotten to the point now where we're able to have pretty significant data and analysis, just given how difficult the logistics of it are. So really the thing that is most important about this trial is this fact of multi-environment testing. So multi-location testing is a pretty common thing in agricultural research. You want to see how different genetics perform in different environments. What's really unique about the International Multi-Location Variety Trial is just how many environments we're covering and how many countries. A typical multi-location trial for corn, for example, might happen in 3 counties in Iowa. And here we're talking about 29 sites in 18 different countries. So it's tremendous diversity that we're getting. And you can see in this photograph, you can just kind of begin to get a sense of some of what that diversity looks like, right? Very different environments, the sort of deeply shaded agroforestry environment here in India, down in the bottom right, to a very kind of hot, dry climate in our Zambia trial on the left. And the reason that we do this is because we want to understand how these varieties, which aspects of these varieties' performance is stable across environments. So regardless of what the climate in that particular location is, are they performing well, or how much of that performance is tied to the specific environment? And from that, we can learn a lot, and we can do a lot, and researchers can use that information in order to, for example, make selections for future breeding and crosses. So just to give you a picture, a feel for some of the diversity that we're capturing here, you can see we have taken all of our sites and clustered them. So we do a little analysis to see which sites are like and different the other ones. So on the left, you can see we have four main clusters. On the right, you can see we've basically done a similar thing, but we've categorized all of the sites according to some agroecological zones that were published in a paper, hot and dry, cold and variable, hot and wet. And you can see that we've done a pretty good job of matching the diversity of sites in the IMLVT trial network to the diversity of agroecological environments that exist overall for Arabica production. So it's not exact, but we have pretty good representative climate variability in our trial, meaning we can draw some good conclusions from the data in this trial about performance overall. So I think I'm going to turn it over now to Tanya. No, sorry, I'm going to do one more slide. Just to show you an example of why this matters so much and why taking these varieties out of maybe the environment where they were originally created and putting them in new environments matter. So in 2017, in one of our sites, we had a really significant frost event. The plants were still young, so they were even more vulnerable than they might have normally been. And you can see on the picture on the left, you have really like rows of coffee that are basically just completely dead, but you can see a little bit of green. And so basically what happened was that of the 31 varieties in this trial site, most of them died, but a few of them did not. It was a couple of varieties from Brazil and a couple of F1 hybrid varieties. And that was really fascinating learning for us because the Brazilian varieties weren't so much of a surprise. Brazil has bred for frost tolerance for many years, but the F1 hybrid varieties in this trial were developed in Central America where frost really isn't a problem. And so even though we would expect them to have overall good vigor and good performance in a climate variable location, we didn't know for sure that they were frost tolerant until we had this result. So when you put these varieties in all these different environments, what you're doing is exposing them to a lot of different stressors. And we're essentially using the stressors and the variability that we get from this spatial distribution, right? Lots of plants in lots of places to help us approximate the kind of variation that we're going to see over time, temporal variation with climate change. We're using the variation we have now around the world, which is really big when you have a global trial site. 28 sites in 18 countries is a lot more variability than you would get if you were only testing varieties in Guatemala, right? Which is a small country with not that much climate variability. So we're putting them in a lot of environments and getting a lot of variability. And that helps us understand how varieties might perform when we have increased variability in the future because of climate change. So that's the relationship between multi-location testing and understanding climate resilience. And now I'm going to turn it over to Tanya, and she is going to walk us through some of our results.
Great. Thank you, Hannah, for such a nice introduction there. So I'm going to take you through a little bit more of the technical detail about this trial and show you some of the results that's coming through. And actually, it was quite challenging to put this together because it's such a large trial, such a big data set. And Jorge handed me a whole lot of different charts and graphs and numbers, and trying to sort of sift through that and come up with something that tells the story was quite challenging. So hopefully here I give you a bit of a taste for it, but certainly understand that there's a whole lot more there in this data set. It is truly a valuable, unique data set, and we're really excited about what we can get out of it. So I wanted to start here with, kind of the timeline of the trial. This was designed as a 6-year trial. at time 0 is when the plants go into the field. And then really we evaluate for 6 years. The first 3 years is really just the plants getting established and growing. So they're not yet yielding coffee, but we can still evaluate their growth and look at their response to disease pressures. But then the second batch of 3 years, so years 4 to 6, is when we really start to get the valuable data. And this is where we can get some good yield data and start to look at things like cup quality. So this was how it was designed, as a 6-year trial. And then once we get to the 7-year point, that's where we have to start to think about, okay, what next? Do we close the trial down? How do we transition to something else? And what do we do next? So that's kind of where we're at for a lot of the sites. And this slide here shows that 6-year timeline and how it relates across all of the sites in 18 different countries. So you can start to see some of the complexity of this trial, that it didn't all start at the same time. It's not a nice, neat, everyone say go at the same time. This was a huge, ambitious undertaking, which, I give a lot of credit to the people who were involved at the start, who designed this trial and negotiated all of the variety access and the partnerships. There was a lot of work that went into the setup of it, and not all the partners came online at the same time. So you can see that in this, how all the timelines are staggered, which means as the data's coming in, it's all at a bit of a different stage, and we have to analyze it and package it together. It gets a bit complicated. But what you can see here is in 2022, so this is where we're at right now, we really are kind of at the peak where we have a large number of the sites reaching maturity. So this is where all the data, the valuable data is coming in. We've had a good amount of time to collect that data, and we have to start thinking about what does that next phase look like. So that's where we're at now. And so that's why it's an interesting point to start talking to you about it, because we can show you some of the results and then start to think about what else can we learn from this and what are we going to be doing next. So when we talk about this trial, we talk a lot about performance, plant performance. And so what does that really mean when we talk about that? It's performance really means a whole bunch of different things depending on what you look at. So we think in terms of the traits of the variety, whether you're evaluating varieties or breeding, you're always thinking about the quality, the traits, the target traits that you're trying to improve. And so in coffee, we're really thinking about 3 high priority traits. So first and foremost, you think about the production at the farmer end. So you think about production traits, which are yield. It's the thing that matters most to farmers, is you have to maximize the yield for them to maximize the value of what they're getting from their production efforts and their land. So yield is always number 1 for a farmer. Disease resistance follows shortly after. So every crop on every farm is beset by different pests and diseases. And one of the best tools you can have to fight disease is by having resistant genetics that doesn't succumb to diseases. So that's a real high priority for any variety. But then when we think about coffee as a highly specialized, high-end consumer product, we also have to think about consumer traits. And so this is where cup quality comes in. So we look at a combination of production and consumer traits. And really, these are the top 3 things that matter. These are the biggest, most important things, and the universal things that matter to every country, every coffee-producing country, and every researcher and breeder. And these are the things that everyone has in common. They're not necessarily, of course, the only important things. There's a whole lot of other things you could look for. But when you running a trial as large and complex as this, you want to kind of focus on really those core, try to simplify it, focus on those core priority things. And so that's where we've centered on these 3 traits. And so all of the performance results that we show is really about evaluating according to these 3 things. So I'm going to sort of walk you through them one at a time. So when we look at yields, overall, across all the sites, when you average the yield data, you can kind of measure it and average it and simply rank all the varieties according to which ones yield the most. And so this is that result. So in this chart, you've got the yields on the bottom axis. So as you go to the right are your higher yielding varieties. And so you can see the top scorers there are EC16, H1 Centro Americano. These are the highest yielding varieties, which also happen to be F1 hybrids. And so if you followed some of WCR's work and one of the previous presentations that I gave, it was all about F1 hybrids being high yielding, high performing varieties. And so this is frankly not that surprising to see that they really top the list in terms of overall yield. And then down the bottom end of the scale, we have some lower yielding varieties, like some of the older material, like K7, Pacamara, as well as Geisha, which is well known for quality, but the yield performance isn't really there. So that's the overall global results. But what you see is it starts to get interesting when you zoom in and you start to look at the results on an individual site and environment basis. And so this is the yield data presented in a different way, where we have every point on this graph is yield results for a different variety at a different site. So along the bottom, we have all the different sites. So our 29 sites and the performance of all of these varieties. And what you can see is the lines go all over the place. They are not consistent. Each variety does not perform the same in different sites. If that was the case, we'd just see a nice bunch of parallel lines, all the varieties performing consistently in every site, but we don't see that. It's all over the place. And so if we zoom in on our highest performer, EC16, that's the bolded orange line there, you can see it's a really high yielding variety in most of the sites, but not all of them. So if you look over to the left here, you can see IPR 103 at that particular site is actually even higher performing. So that at that particular site, maybe IPR 103 is the variety you would perhaps favor. Similarly, at the lower end of the scale, you see Pacamara is, fairly low overall, but not in every case. So this starts to show you how the variety and the environment start to interact, and the performance is different. They're not always consistent across all of the sites. And so this is what we, in genetics, we talk about the GBYE interaction. So genetics and environment interact together to create the plant's phenotype, which basically means its performance. It's the way it visually appears in the field. So a plant's phenotype is a product of genetics and environment, and we say there's an interaction between these two things. So we call it a GBYE. So that sort of signifies the fact that those two things interact. And so what we can see by looking at the data is sometimes, like in this example on the left, we have consistent performance between sites and varieties. So on the example on the left, we have two varieties, EC16 and IPR 107, at two different sites, and they consistently perform in kind of the same way. EC16 is clearly performing better than IPR 107, the same type of response at each site, even though the overall yield might be different, the pattern is kind of the same. And so on the right, we have a different example where we're comparing IPR 103 and SLN 6 at two different sites. And this is where you can see at site 1, IPR 103 is a higher performer, and at the other site, it's the other way around. SLN6 is a higher performer. And so this is an example of where we are seeing this GBYE interaction, where the genetics of the variety and the environment are interacting in ways that you don't necessarily predict, that it sort of switches the results and it creates this different performance. And so that's what we mean when we say GBYE, genetics by environment interaction. So that's just the yield data. When we zoom in on the disease elements of this, the number one thing we're looking at really here is coffee leaf rust. So coffee leaf rust, which you're probably familiar with, is a disease that it basically devastated coffee production in India more than 100 years ago. So that's where it arose first, and since then has spread to nearly every coffee producing region in the world. And so this is one of the top diseases in coffee, and really a strong target for developing resistant material. And so the way we look at coffee leaf rust is we can evaluate the performance of the variety in terms of this scale of severity. So it's kind of a standardized way of looking at the tree, counting the number of spots and evaluating the level of disease severity and giving it a score. And that way we can analyze this data. And so what that looks like is this. So a similar chart as to the one you saw for yield, except in this case, higher scores are worse. Higher scores on the right-hand side mean more severe disease, and this is not what you want to see. And so again, when we average across all the sites around the world, what we're seeing is the highest rust incidence is happening in some of these varieties at the top. So Pacamara, SL28, K7, those are some of the most susceptible varieties to rust. Down the other end of the scale is where we have the most resistant varieties. And here we're seeing EC16, Riru 11. These are more resistant varieties. So this is really valuable information. And interestingly, if you remember the yields chart, the same two varieties, Pacamara and EC16, are at both ends of the scale for yield and for rust. So this sort of raises the question then, is the yield response we're seeing, is that a result of the rust pressures? And so there's, different ways we could look at the data to answer that question. Another element of disease that we're looking at is coffee berry disease. So this is another severe disease of coffee, but instead, this one isn't a global phenomenon. This one is present primarily in Africa. And so what that means is we can't really do a global analysis of coffee berry disease because it's just not there in Latin America and other sites around the world. So it's something we can only evaluate in Africa. But of course, there's always the risk that these diseases can spread around the world. So partners in other countries outside of Africa are still very interested in seeing the results of this work because of the potential risk of this disease spreading to other countries. As well, some of the variety owners and the people who've developed the varieties in other areas of the world, so we have some of the EC15 and 16, for example, have been developed in Central America, and so they wouldn't have been exposed to coffee berry disease. So it's very interesting to see those varieties now being challenged with this new disease to see how they perform. So in this chart here, you can see it's a kind of a different way to present disease data. What we have in all the columns up and down are all the different sites in Africa. The varieties are there listed on the side. And if you look across the rows, there's little blue and red squares. Everywhere there's a red square, it means the disease is causing a problem. It's highly susceptible, and the blue is showing less disease or more resistant material. So really what you want to look for are the varieties that have more blue squares. And in this case, it's this sort of cluster down at the bottom where we're seeing more resistant material to coffee berry disease. So some of those varieties are doing really well in the African countries. One of the standout observations you can see here, though, is the variety Oro Azteca, about 2/3 of the way down there, is pretty much all red. So this variety was developed in Mexico. It wouldn't have been exposed to coffee berry disease. But as we see in Africa, it's not doing well. So it's highly susceptible to this disease. So if you're in an African country, you're probably going to think Oro Azteca is not the right variety to be introducing there. And so the other thing you can see from this data is all the little crosses, the absent missing data. And this is, reflects one of the challenges of running a trial such as this is for various reasons, the data can't be collected, it's not done in time, or it's incomplete. And so we do have missing missing data. And so over time, we can improve this as we get data in from multiple years. It reduces the problem of missing data. And so we can start to fill in some of those gaps and have greater confidence in the results that it's showing us. So one of the things we think about when we think about testing varieties or genetic material in different environments is what we call the discriminating capacity of a site. So what that really means is how well does this site differentiate between different variety performance? And so I've picked a couple of examples here to show you where we have 2 test sites, one in India and one in Nicaragua. And if you look at yield, so these are little small reduced down versions of those same types of charts, which show yield data and the varieties ranked. So in India there on the top left, you can see all of the varieties essentially yielding fairly low. And so under these kind of conditions where everything is performing low yields, you're not seeing a lot of differences between varieties. All the varieties kind of look the same. They're all fairly low yielding. But then when we look at Nicaragua, moving across to the right, this is a much more optimal coffee growing environment where it really starts to maximize the potential of the trees and the yield potential. And it really starts to differentiate between the varieties in these optimal conditions. So you can see this much nicer spread of data between the higher yielding varieties and the lower yielding ones. And so Nicaragua has great discriminating capacity in terms of yields. But then when we look at rust, it's the other way around. So you can see in India, we have, as I mentioned earlier, India has the highest rust pressures of any place in the world. So there's a lot of rust pressure, a lot of different races of the disease and high pressure, which means it's exerting a lot of pressure on the varieties, and the performance and their level of resistance starts to become really differentiated. So the data that we get from India is really good for identifying the varieties that are most resistant to rust. Nicaragua, the opposite way around. Not much rust pressure there. And so it's really quite hard to tell if one variety is doing better than the other in terms of rust resistance. So this starts to show you how we can use the sites and the data and all of this global analysis to really get a good description of the performance of these varieties in each of the different environments. And then the third piece we're looking at is cup quality. And as you can imagine, with 30 odd sites and 18 different countries, trying to cup all of that material is a real logistical challenge. And so we handle this in a couple of different ways. We do local cupping. So these are the partners in the countries. They do cupping on the material from their own sites, which enables them to see how the material performs in their own environment. But for us, trying to centralize this data and look at global performance, we need to be able to compare the performance across different countries. So we can't necessarily compare different cupping results unless it's been done in a more centralized way. And so this is where we have developed what we call our GBYE cupping experiment, where we've essentially taken a subset of this material to do cupping in a more centralized way. And so what that means is locally at each of the origins, the material is processed, and then our staff box it up and mask the labels so no one knows what they're getting, and then divide all that up and send it off to different roasters. So each company that receives this material roasts it according to their own practices and then does the cupping themselves. So they're evaluating a mix of this material across all the different sites and the different varieties and the different sites using that same standard cupping protocol. And so this is a way for us to have a look at the global interaction of genetics and environment in terms of cup quality. And so right now, what we have is really just the first year of this data. It's the 2021 data that we have that I'm going to show you, because the 2022 year is currently underway, and some of you will be involved in that cupping process. And then we've got one more year after that, which we'll be doing next year. So these are some early results that we have here. And so again, when we look at the global overall scores, you can see that some varieties overall do perform well in terms of cup quality than others. So you see Marsellesa and Batian are at the top of this list in terms of cup quality. Down the other end, H1 Centro Americano is lesser quality. And Pacamara, as we've seen in some of the other slides, is somewhere in the middle here in terms of cup quality. But again, when we zoom in on individual sites, you see it's not consistent. You see there's this differentiation between different varieties performing at different levels at different sites. And so Pacamara, in this example where the arrow is showing, that's actually the highest quality performer at that particular site. And so again, you're seeing this genetics and environmental impact, and different sites, different varieties perform differently. Another interesting thing to do is to look at the correlations. So when you have, for each of our sites, we have all of the environmental data, the altitude, temperature, rainfall, and we can correlate that with the cup quality scores and start to see some of these trends. And so one of the things that's widely understood is, coffee grown at higher altitudes is higher quality. And we do see that in the data here. So the chart on the left in the blue shows you at increasing altitude, you're getting higher cupping scores for the coffee that's being tested. What we see here also, which is perhaps less widely known, is you see similar correlations with temperature and rainfall with cup quality, except it's the other way around. So as the temperature at a site increases, it actually reduces the quality scoring that the coffee gets. Rainfall, same story. So the more rainfall that a site receives, then the lower quality the coffee would be from that site. So this is a really interesting trend to observe, and certainly something that gives you some insight and pause for thought when you think about climate change and the way some of our coffee producing regions may be changing over time in terms of the climate that they face, and what effect is this going to have on the quality of the coffee that they are able to produce. So that's just a really sort of high-level short taste of some of the data that's coming through. And the way in which we're using this is kind of fourfold. So the first most obvious thing is sharing it with our partners. So this huge trial involves all different partners who are collecting the data and very interested in seeing how the results in their country measure up to other countries. Sorry, just got my little pop-up there. And so we share the data with them, and they are able to look at the performance in their own country, as well as many of our partners are also contributors. They're variety developers. They've contributed some of the varieties to this trial, and they're very interested in seeing how their own varieties perform in other countries around the world. So everyone has kind of their different perspective. They're very interested in the data, and they look at it to answer kind of their own questions. And so we have that's an ongoing conversation that we have with the partners in the trial. The second thing is we want to make this public. Right now, it's a kind of a closed trial because we have signed agreements with all of our partners to share this data. But ultimately, we want to publish it. We want to make it public because this is how we contribute to the overall knowledge and understanding of coffee varieties, environmental factors, performance, genetics, all of those things. It helps contribute to the body of knowledge, and it releases the data to allow other experts, other scientists to dig around in the data and start to answer some interesting questions of their own. So that's really our goal. We want to get there. Once the data has all come in and analyzed, we do intend to publish it and release it publicly. And so right now we're just starting to explore some of those things. Is what are we going to do with all this data? How can we really extract the value of it? And how can we share it? How can we mine it to answer specific questions? How can we share it? Who do we share it with? All of those things, conversations that we're having right now. And then the fourth piece, the fourth way in which we use this data is really, it's in the business of science. As soon as you find get new data, something interesting, it just creates more questions like, oh, isn't that interesting? Why is that happening? So there you've got another question, you've got another research project, and the data informs future research. So it kind of feeds the engine of science and informs the next piece that we may want to do. And, this is another reason why we want to release it publicly, because we can't do all that ourselves. We want to release it and let others add it and see what they can find out of the data and what sorts of future trials and experiments it could be used to inform. But if we think internally at WCR, the way this data has informed already future research is what Hannah mentioned at the start in the launch of Innovea. We've just launched our own breeding network, but really this whole breeding effort and the network that we built for breeding our own varieties was really rooted in this giant IMLVT trial, because IMLVT allowed us to bring together all of those different varieties and really understand which ones have the traits of interest, the highest performers in terms of yield and disease resistance and cup quality, and then start to think about how can we incorporate that material into a breeding design to get the best of all of those traits into even further improved varieties. So this data has been critical for that, but more than that, it's been a way for us to build the research partnerships and these collaborative relationships we have with all of the partners on IMLVT have really morphed into deeper relationships under Innovea. So a lot of the same partners who've signed on to the Innovea network were working with us originally as part of the IMLVT trials. And so it's kind of a logical evolution of it that is now being launched into Innovea. And really, a lot of credit is due to this original trial that was set up 6-7 years ago. But back to the IMLVT itself, as I showed in the timeline slide, the data coming in is really at its peak right now. And it's time for us to start thinking what's next. And the way this was designed is it's kind of like a funnel. So at the outset, we had a huge number of countries. All of this data is coming in. We had sites. Some of the sites didn't perform so well. Some are less value than others. And so we're starting to think of it as a funnel, a narrowing down process. So we take this huge, gigantic trial, and over time, we narrow down the number of sites we're working with. We do need to reduce the overall footprint of the trial. It's a very large and complex thing to manage. And as we start to think about closing it down, we need to think about how can we focus our resources on really the highest value pieces to really maximize the value of what we're getting out of it. Because what we are seeing is there's a really strong demand for this. This is a really global, unique trial that has never really been done on this kind of scale in coffee. And so there's a strong demand for the results and for this trial to even keep going. So our country partners are eager for new varieties. They don't want to close the sites because they want to evaluate the performance over a longer period of time. We also have member companies who are interested in the results because, they have their own questions about which varieties, which countries, which quality, all of these things. And so you can start to answer some of those questions by digging into the data. And as well, as I mentioned earlier, the scientific value of this global data set is increasing. The more data that comes in, the more years of data that we have, it's really enormously valuable in generating new types of information that we're starting to see. So we're kind of in two places. We're concurrently trying to reduce the footprint and the management complexity of this trial by closing some of the sites, as well as think about how could we retain a core of really valuable material, and think about how we can get the value out of it over time. And so we're looking at kind of two streams: is what could be a piece that we might want to continue, so we have a more longitudinal data over time. And then for some of the partners, they've already identified these are the top performers in our country. We want to move that out towards commercial production in our country. And so that's where we need to think, okay, what does that involve? That's a whole different process of moving it out to pre-commercial trials, on-farm trials, evaluating it at a larger scale within one country. And so that's another thing that we're exploring and working with some of our partners on. So there's a lot of things happening with this trial overall. It is evolving. And so, it's a really interesting time to be a part of this. So that's really the end of my presentation. But before I end, I really wanted to pause here and acknowledge, the involvement of all of our partners, because it is such a huge, ambitious trial. It's a global effort, and it's really, all of these partners, these are the ones who are managing the trees, collecting the data, sending it to us, and we absolutely could not do it without them. We're a very small organization operating through this network of partnerships, and so a lot of credit really needs to be given to these partners, and it's because of them that we're able to produce all of these interesting results. So then I'll pause it right there. I think we have some time for questions, maybe 10 minutes.
Yeah, we do. So folks, please feel free to type your questions into the Q&A box or the chat, either one. We'll keep an eye on both. We can always scroll backwards and, take a second look at any of the slides if you have particular questions about those. Maybe I'll just, while we're waiting for some to come in, I'll throw a question out. I'm always very intrigued by the kind of behind the scenes piece of how this works. Like, how does science actually get done? And Jorge, since we have you on screen here as our kind of data analyst, I'm just curious to get your reflection on, like, what is it like to work with all this data coming in? It's coming from all these different people, all these different places over different years. Every trial's in a kind of different state. What sort of challenges does that pose for analysis?
Well, it's a lot. So it's a lot of data. That's one thing. A lot of data. So if you think, and especially, for example, let's say for rust, at the beginning we were measuring each tree separately, instead of the 10 trees, like kind of like an average of the 10 trees per. So if you think, like, each site has 31 varieties times 3 blocks times 10 trees. Then it's just one site, and then you cannot. Then it's 20 something sites. So that is a lot, and also one of the issues is just looking for typos. That always happens. A 1 becomes a 10. So and I mean, everybody does those. I mean, I make those mistakes too sometimes. So so kind of like go through different cycles of quality control, and that's and kind of compiling. Also, the issue that you mentioned about the sites starting at different years. So how to compare sites that have 3 years of yield versus 2 years of yield, and how to account for that. So that's I mean, it's a challenge, but you can actually make some cool findings with all this data.
Awesome. Tanya, question for you. So you kind of came in, you joined us not even a whole year ago. So this trial's been underway since 2015. I'm sort of, and you came from other crops. Coffee was not the crop that you had worked on prior to this. So I'm sort of curious to hear your reflection on just this trial overall. Like, how does this multi-location trial compare to other kinds of multi-location trials that you've seen? in either its complexity or global nature or anything like that. Can you kind of just help us orient to like how to understand the scope of this trial?
Yeah, I think you actually explained that very well, Hannah. I think typically in commercial crops, it's done in a much more focused way. I'm thinking of an example of apple breeding, that I was involved with previously. We're breeding for one country. And so we're focused on a really narrow range of traits and performance criteria. And while there's different environments within a country, it's a lot more focused and limited than what we have here. So here we have such a diversity of different environments, as well as a complexity of partners to work with, who all do things in a bit of a different way. And so there's elements to this trial that's not even really visible in here, because things like the production practices are different in different countries. And it makes Jorge's job more complicated to try and draw conclusions out of this data, because there's kind of noisy elements to it. So this is much more larger and scattered and has an enormous diversity of environments, and it creates a great opportunity to pull out some interesting conclusions and observations, but at the same time, it is much more complex and to navigate some of that variation. And so that is definitely unique compared to the more controlled way it is done in other commercial species that are particularly focused in a particular production environment. So I think, the complexity of it is really astounding to me, but it's exciting as well. I think there's really a lot of things we can pull out of this data that could be really interesting. So I think it's really an interesting time to be joining, because all that complicated setup has already been done, and now we get to see the results coming in.
So there's a question from Tom that kind of touches into exactly what you were just saying, and he's wondering, how much do the variations in cultivation practice or harvest practice or processing techniques, when we're talking about cup quality analysis, how much does that variation influence the outcomes? How do they get standardized or not over the locations? How does that work?
Yeah. Jorge, do you want to take that one?
Yep. So that's a good question, and so that's one thing. The one is that we're not trying to figure out management questions, what works best or not. And the idea was that we could standardize the management to some level, but more closely of what's locally done. Mm-hmm. Because we don't want to evaluate something in, let's say, like like a very high fertilizer management or something like that it will never be a reality in that in that specific country or whatever, so it's actually we don't it's hard to tell apart the side effect versus the management, but that's some of the noise that we have to work with. Mm-hmm. Actually,
it's like another way of saying that is that we're sort of considering that management differences are almost part of environmental differences. They're so at a global scale, to some degree that works, right? You're just looking at across all the variation, where do you see genetic performance excelling or not excelling? Well, I'm sure it presents a lot of difficulties for Jorge, but broadly it works. So it's similar to the processing question. Like, we don't send people a particular yeast strain for their wet fermentation. We're considering that whatever is in the air, we do ask folks to do wet fermentation, so that part is standardized, but whatever the microbial environment is, that just is part of the trial. We're not trying to rigidly standardize in that way, because it would basically be impossible.
I think one of the ways to think about it is there's a bit of a trade-off here between the two different goals that are in this trial. So we're interested in this big global analysis to understand varieties, performance, and effective environment. And if we were designing it with that in mind, we would absolutely want to standardize things and reduce some of this noise so that we get some really strong data coming out of the trial. But what our individual partners are interested in primarily is which varieties perform best in their country. And so, in an example like production practices, we might want to standardize it for everyone, but within a country, they're going to want to do whatever's standard in their country, do things the same way so that they can see how the varieties perform under realistic conditions. So that's a bit of a tension that exists that had to be dealt with in the trial design and affects the way, you think about how the data is analyzed.
Can I just add something? So just also related to this and the questions, just two questions from Santos and from Dr. Lance about, the integration of data and climate. So one of the next steps now is that we're getting climate data from basically the start of the trial up to this day. And the idea is to actually use different climatic data, different climatic variables, to try to draw some conclusions about what of these climatic variables are affecting different traits, and also, for example, how different sites, maybe in different countries, in different continents, could be actually having very similar climatic conditions, maybe different soils or something like that. But all this variability and between sites could be studied a little bit more with just climatic data.
We have time for just one more question. And there's a great one here that I think gets at one of the other important purposes of the trial. So, right, one big purpose is just knowledge, scientific information that we can use for future breeding efforts and to understand variety performance. But the other is the way countries are using this data to evaluate whether or not they may have interest in releasing some different varieties in their countries. So Christopher asks, is it likely that some of the new or improved varieties might be specific to geographical regions? And I think what this question is beginning to get at is, like, how might countries use this information to think about expanding the options for what varieties are available in their countries? Tanya, could you speak to that a little bit?
Yeah, so that absolutely is. I think over time we've come to understand that the partners, that's the thing they're interested in most, is they want access to new varieties above and beyond what they already grow. And so this was a great opportunity to get new and different varieties from all over the world into their country, so they can get some experience and evaluate under their conditions. So many of our partners have identified some of the varieties. This is new. We don't have this yet in commercial production. It's performing well. So can we bring it in? And so this is something that we are working with them, and it has to be done on a case-by-case basis, because it's specific to each country, as well as the accessibility of the variety. So some of these varieties are owned by other groups. Some of them are public domain, but some of them are owned. They are restricted on the ways they can be used, and it all needs to be negotiated. So it needs to be negotiated country by country, variety by variety. We're trying to help where we can with that process, but ultimately it has to be a bilateral agreement between the country that wants to introduce the variety and the owner of that specific variety. And so all of that is a process that, is kind of starting to happen now.
And it may be, and in fact, we're seeing with some of our partners that different countries are interested in different varieties because local performance is different. The highest global average performance may not be what's best in a given country in terms of yield or quality. And so partners are looking very closely at their specific data, their specific results, to see what's going to work best in their location. And it's not going to be the same variety in every place that's performing sort of best according to the three metrics that we have. But country partners are also looking at their own, they may be collecting extra data and thinking about it through the lens of what's going to work for our market, what's going to work for our farmers. So I wish we had, another hour to answer every question and all of that, but we're at time, and I just want to respect people's time. We want to thank you all so much for joining us this morning. We are talking about having an additional kind of deeper dive for folks who are interested. So you should have seen a poll question about that. We will reach out to those of you who said you were interested to look at scheduling some additional conversation. We have some information up on our website. We are going to be bringing you more from this trial over the coming months and years. This is just the beginning, not the end. So thank you so much for your attention and your great questions. And we look forward to seeing you soon next time.
Great. Thank you.
Thanks.